in epigenetics or other fields of, that are related to the subject of the talk. But if you are more interested, if anyone in the audience is more interested in additional details or to ask more mechanistic questions, so don't hesitate. We can do it in the end of the talk or during, whatever you want, okay? So let's see. But we'll start from very broad perspective and we'll see how it goes. Okay, great. So the subject of the talk, we work on many different things in the lab, but today I will discuss heritable memories. The possibility, which sounds wild in the beginning, that some memories can be transmitted across generations. And I will start by saying that we know that some memories are innate or encoded in the heritable materials. We know this. This is something that was discussed already by the Greeks thousands of years ago, but we also know it from different types of research, more or less anecdotal. For example, in the 80s in the United States, there were experiments that were conducted with monkeys that are born in captivity. So these are researchers that work in psychology, and they studied these monkeys that are born in the lab, so they don't know anything aside from what the experimenters showed them. And they found, these researchers, that it's very hard to teach these naive monkeys that toy flowers are dangerous, that they are scary. So if you pair a toy flower to something scary, like an electric shock or a gong or something like that, they won't be afraid of it. You need to pair the toy flower multiple times with something scary until the monkeys actually realize that this is scary, that they will learn this association. But it's very hard to teach them. On the other hand, while the monkeys are not born with the fear of snakes, it's very easy to teach them that snakes are scary. So if you take a toy snake and pair it with something like an electric shock, the monkeys will immediately learn that snakes are dangerous, and they will learn to fear snakes for the rest of their lives. So they were born with a capacity or tendency to be afraid of toy snakes or snakes and not flowers. The question is, so we're not born a blank slate, but the question is, where is this memory encoded? It's definitely not in synapses, which normally when we think about memory in the brain, we think about memories that is stored in synapses, these connections between neurons. But we know that this memory, this innate memory, is not stored in synapses, but because we are all born from a fertilized egg, a single cell that by definition, since there aren't any other cells around, doesn't have any synapses. So it has to be encoded in some molecules in this original cell. And the question is, can we characterize it? The problem is that memories are very mysterious. We don't know enough about how memory is encoded in the brain and how genetic information translates to memories. We just don't know anything about it, and it's very hard to study, especially if you study humans or mice or organisms with complex brains, because our brains are just immensely complicated. So we have many billions of neurons, and each of them is connected by thousands of synapses to other neurons. So just the complexity is out of this world, really difficult. Which is why in my lab we to simplify the problem, we focus mostly, not always, but mostly on very simple organisms or organisms with a very simple brain, and we study C. elegans nematodes. And these worms, which are microscopic and very simple, have just 302 neurons. That's it. Always 302, not 303 or 301, always 302. And we know also all the synapses that these neurons make. So we know that altogether it's less than 7,000 synapses. And despite the enormous simplicity or the reduction in complexity that exists in this worm, the worms are still capable of doing very complex things. So obviously, unlike us, they can't write poetry or tweet or stuff like that, but they're very smart as well. So they sense thousands of odors. The worm encodes for something like 1,300 different GPCRs, and they can mate, which is complex. They can sense temperature and touch, and do really complex things, although their brain is so extremely simple. And the most interesting thing that they can do with their brain is learn. They can learn very simple associations. For example, an association between odor, which is how they navigate in the world, and food. They can learn that certain odors are attractive, or they signal that there's food around or not. We also have a map of the connections of the brain of the worm. This is a connectome. It's the only organism that has a completely deciphered connectome, and we have this map of the brain at the resolution of the synapse since the 80s. So we really know each neuron in the worm by its name, and we know how to get to these neurons and how to change their activity to control the worm. We can operate these worms like a remote control car. We can, using optogenetics, for example, shine light on the worm and make it move forward or backward or lay an egg and so on. So it's a really impressive level of understanding. And despite how well we know the nervous system of the worm, we still don't know anything, many things. So we don't know more than we know. It's also time to be a little humble with our aspirations to understand the human brain and so on, if we can't really figure out this extremely simple nervous system. Okay, I wanted to study memory. So I went to study C. elegans, but moreover, I wanted to focus on a very specific and well-defined memory. And this is a memory that's called imprinting. So when I went to do my postdoc, that was 10 years ago at Oliver Roberts' lab, I wanted to study imprinting. And imprinting is a type of memory, a long-term memory that you can only acquire during a very specific stage of life, normally early on in development. So for example, you probably heard that salmon, that these fish, they swim back against the current to the place where they were born. And they do this by searching for familiar smells. Similarly, neonatal attachment is a strong bond that is formed between the baby and the mother. And this is, again, a very strong bond that is very long lasting. And also, the phenomena of imprinting was studied by Konrad Lorenz, who studied it in birds and got the Nobel Prize for that. And he showed that birds are imprinted. They form a bond with the first moving stimuli that they encounter in life, and they just follow it around. Normally, it's their mother, the bird's mother. But as you can see in this clip, it can also be something very different, like a dog or a kid. Also in C. elegans, this is my postdoc advisor, Oliver Robert, together with Jean-Jacques Remy from France, they published this paper in Science that also worms C. elegans can be imprinted. Normally, worms forget very fast after 2 hours. But if you make an association between a neutral odor that doesn't mean anything to them, it's not attractive, it's not repulsive, you take this neutral odor and you pair it with food to make it attractive. And if you do this pairing in a specific developmental stage, at the L1 stage, the first larval stage of the worm, then the worm will learn that the neutral odor signals that there's food around, and it will not forget it for the rest of its life. And the worms live for a few weeks. So only if you pair it in L1, in this particular stage, the memory is long lasting. And this is what I wanted to study, because here you have the potential to actually understand how memory works, where it sits in the neuron, in which neurons, in which molecules, how does it work? The problem was that when I got to my postdoc in 2010, Oliver Roberts' collaborator, Jean-Jacques Remy, published another paper which was even more shocking. But he published this current biology paper, and this paper has only one figure, the one that's in front of you, this scheme, so no additional data. What Jean-Jacques Remy claimed in this paper is that the memory, the imprint, transfers also to naive worms that never experience the pairing for multiple generations. And this is very bizarre. This is just a sign of shock. This is bizarre because but you know that what I'm telling you now your kids won't remember it, right? This is obvious, and this is very unintuitive. Not only is it unintuitive, it also goes against some very basic principles in biology. In fact, it goes against a principle that's also known as the second law of biology, or the Weismann barrier. And August Weismann, which was a very famous and important biologist working in the 19th century, formulated a law. He didn't use these words that are written on the page, because on the slide, because these words didn't exist. But what Weizmann said was that, in other words, the genetic information cannot pass from the soma to the germline. The only tissue that can transfer information to the next generation is the germline, the germ cell, the eggs and sperm. What happens in the soma stays in the soma. And since and he formulated this law of his, the Weizmann barrier, this segregation of the germline from the soma, to make Lamarckian inheritance impossible, to show that inheritance of acquired traits that I will not elaborate on, it cannot happen, that it's impossible. And the reason that it's impossible is that the soma, the cells that actually interact with the environment, cannot transfer what happens to these tissues to the next generation. So if you go to the gym and you build muscles, it doesn't matter that your muscles change because it doesn't affect your germ cells, which transfer the information to the next generation, because the next generation will be formed only from the sperm and the egg that will later divide and define the other tissues in the body. And this is considered to be completely correct. Inheritance of acquired traits until recent years was considered to be something you just can't say to a biologist, a forbidden thing. And the reason that it's such a dogma is that it fits what we know about genetic inheritance, how DNA-based inheritance work. So we know that each cell in our body has the same DNA, and that if you have a mutation, for example, or any other change in the DNA of a somatic cell, let's say you have a mutation in your hand, there's no mechanism that will transfer the same mutation to the DNA of the germline. So it doesn't matter. For the next generation, it doesn't matter whether you have a genetic change in your soma, right? This is why inheritance of acquired traits can't work, and the Weizmann barrier is considered to be correct. Moreover, also epigenetic changes, so heritable changes that are not encoded in the DNA sequence, for example, chromatin modifications, are largely erased in the germline and early embryo. So there's a program that's called a process that's called reprogramming, or epigenetic reprogramming, which resets histone modifications and cytosine methylations, so that the next generation will start as a blank slate. And most of these marks are lost during development and in the germline and early embryo. Not all the marks are lost, so about 10% are still there, but by and large, there's a big resetting that reboots the genetic material. So it looks like inheritance of acquired traits or inheritance of epigenetic memory is impossible, if that's the resolution, if that's what you're looking at. And this is true. However, it seems that worms, at least, and RNA doesn't care much about the Weizmann barrier. So we know for 20 years, since the Nobel Prize worthy discovery of Fire and Mello in C. elegans nematodes, that RNA responses, so RNA interference effects, silencing that is mediated by double-strand RNA and the small RNAs that are made out of this double-strand RNA. We know that small RNAs and RNAi can move between different tissues of C. elegans, and also there's evidence that small RNAs are present in different bodily fluids in humans and mammals. And in C. elegans, we know that small RNAs can move between cells, including from the soma to the germline. So if you inject a somatic tissue of C. elegans with double-strand RNA, you get the silencing in that tissue, but also in other tissues and in the germline and in the next generation. Moreover, you can just feed worms with bacteria that produce double-strand RNA. These worms eat bacteria. And the silencing would spread out from the site of ingestion, from the gut to the rest of the body and to the germline to induce RNA or silencing in the next generation. So that's a clear breaching of the Weizmann barrier. And we know this for 20 years. We also learned quite a lot about the mechanism, although we're still missing many of the details, but we do know that there are at least 8 different genes that function specifically in non-cell autonomous silencing, in systemic silencing, in movement of the signal of RNA between different tissues. The most famous of which is SID1, discovered by Craig Hunter, which is also conserved in humans. However, I'll add that all of this is true only for exogenous artificial double-strand RNA. We don't know whether endogenous small RNAs can also move between tissues, although we do have clues that they do, but we don't know exactly how, and we don't know which small RNA species do it. So a lot of what we know was learned about exogenous artificial double-stranded RNA, and we still need to understand much better the transfer of endogenous small RNAs, natural small RNAs. When I started my postdoc, and I wanted to study this, I said, forget about memory in the same generation, let's study this transgenerational memory. But I knew that this imprinting assay is difficult, it's a complicated behavioral assay. I wanted to ask which type of molecules mediate inheritance of inheritance of memory, and what type of memories can be inherited. And at the time, there wasn't anything in the literature about real physiologically relevant transfer of small RNAs between generations. So I wanted to study it. But I read the literature, I read all the literature. There wasn't any clue about a memory that can be transmitted across generations by small RNAs or RNAi. By the way, people at that point didn't know that what's being transferred between generations are the small RNAs. It was possible that the double-strand RNA transfers, or it was also possible that histone modifications transfer to the next generation, because these small RNAs also move into the nucleus to guide the position of chromatin marks. So nothing was known about that. So after I read the literature, all the relevant literature, and I almost gave up, I did what I think most scientists do, whether they admit it or not, and then I just read Wikipedia. And in Wikipedia, in the part about the ecology of C. elegans, there was a sentence that was since then removed, an amazing sentence. It was removed not because it's not true, just because Wikipedia updates very often, and there was new information. The sentence that was written there in the part about the ecology of C. elegans is that C. elegans is one of the few forms of life not known to have a natural virus. And this is quite amazing because viruses are extremely good at infecting. You know this, and we are experiencing it as we speak. And also it was amazing because there are thousands of labs that study C. elegans. Many have looked for viruses that naturally infect C. elegans and didn't find them. And I knew that RNAi, this RNA interference system, or works via small RNAs, evolved as an antiviral system. So I thought that perhaps since the worms are so good at RNAi, what happens is that when worms are infected with RNA viruses, that just the virus at some point has to replicate, so it has to form a double-strand RNA, which looks suspicious to the worms, and then maybe using this double-strand RNA starts an RNA response, leading to the creation of antiviral small RNAs or viRNAs that lead to the silencing of the virus, and that this is how the worms protect themselves against viruses. And indeed, these worms, they don't have dedicated immune cells such as NK cells or T cells or B cells to fight viruses. They use RNAi, and they are very efficient in doing so. And the hypothesis that I had was that just like with artificial small RNAs, artificial double-strand RNA, perhaps the amazing capacity of worms to protect themselves against viruses is confirmed by heritable small RNAs. So if one worm succeeds in producing a potent RNA response against the virus, it will transmit it to its progeny, which will be born immune. It's sort of a heritable vaccine type of hypothesis. And there were other labs studying viral infections in C. elegans using artificial viruses, because there wasn't any natural virus that infects C. elegans. And they showed that you can take a very simple flockhouse virus, and also other viruses, integrate GFP, green fluorescent proteins, into the genome, and follow the infection. And what they saw is that if you try to infect just wild-type regular worms that have a functional RNAi system, then the worms protect themselves very efficiently against the virus. So you see here on the left that the worms don't shine in green when they are infected with a virus, because they have a functional RNAi system. They create small RNAs against the virus that destroys it. On the other hand, on the right, you can see these green worms. They are fluorescent. The virus propagates in these worms successfully because these worms don't have a functional RNAi system. So these are videos that I made using different RNA mutants. Here is shown RD4, I believe, which is a double-strand RNA binding protein that's crucial for RNAi. So you see, since it can't do RNAi, the virus propagates and infects it very efficiently. So this really links RNAi and antiviral protection and C. elegans. And you can also sequence small RNAs against the virus from these worms that tile the entire length of the viral genome. Okay, so using this system in hand, this fluorescent virus, I wanted to check my hypothesis that small RNAs might be transmitting antiviral memory between generations. So what I did is, I did this simple experiment. I took worms that have a functional RNAi system, so wild-type worms, and I infected them with a virus. Since they have a functional RNAi system, they destroy the virus and they form small RNAs, antiviral small RNAs or virRNAs against it, and you can sequence it. And then in the next generation, using a very simple genetic trick, this is just by crossing, I isolated worms that don't have a functional RNAi system. So these are mutant progeny. So just like in the previous slide, these worms shouldn't be able to protect themselves when they are infected with a virus because they don't have the capacity to mount a de novo RNA response, which is why when you infect them with the virus, you expect them to be green. You expect the virus to propagate, express GFP, and the worms will be green. But that is unless their parents transmitted small RNAs to them that will protect them. They will transfer the immunity to them in the form of small RNAs. So this was a very simple assay that I did. And what I found out when I did this experiment is that 100% of the worms silence the virus perfectly. And you can also sequence small RNAs from these descendant worms, from this F1 progeny. And this is the first demonstration that virRNAs, that small RNAs are actually being transmitted between generations. And the first demonstration that this can actually encode for any physiologically relevant trait. Moreover, since these worms can self-fertilize, we checked also additional generations, and we saw that the immunity transfers to the next generation and also to the next generation for many generations. Further, in this same experiment, I found that transgenerational inheritance of small RNAs against the virus depends on an enzyme which is called RRF1, which is an RNA-dependent RNA polymerase. So a polymerase that synthesizes RNA that uses mRNA or RNA as templates. So unlike polymerases that use DNA as templates, such as POL2, this polymerase uses RNA as template, which gives it the ability to amplify the small RNA response over and over, because small RNAs lead to silencing of the targets and the production of additional small RNAs that will continue the effect for multiple generations. And the worms encode for 4 different RNA-dependent RNA polymerases. Yeah, so as I said, we sequenced these heritable small RNAs and demonstrated for the first time that small RNAs are actually being inherited. We also followed the transmission of the effect between the progeny, and we saw, we didn't characterize it in high resolution, but we saw that it cannot be explained by the rules of genetics, by Mendelian rules of genetics. So it's not DNA encoded. It follows a different set of rules that we'll describe later on, as I'll show you later on in this talk. While I worked with an artificial virus, Craig Mellow's lab, a few years later, this is the person who got the Nobel Prize for RNAi, showed that also with more natural infections with different viruses, the worms produce a transgenerational response, protective response, heritable immunity in the form of small RNAs. So the same thing happens also with other more natural viruses. So it's a general principle. After that, I thought that perhaps there are other memories that can be transmitted between generations, not just immunological memory. And the first response that I wanted to characterize was the response to changes in diet, or more specifically to starvation. And the reason that I wanted to study epigenetic inheritance of starvation is that starvation is the one trait that was most well studied in humans and in different rodents, and shown to have to continue in the progeny. So the most famous study in humans about epigenetic inheritance is the study that was done on the Dutch famine, or the hunger winter, this period where the Nazis in the Second World War starved large populations of the Netherlands for about half a year. And there were food ratios, no one ate more than a certain number of calories, and they were starved for half a year, and people followed the descendants of the starved women, their children and also grandchildren, and saw that their children and grandchildren exhibit all kinds of traits which cannot be explained based on just genetic changes, such as a propensity to develop diabetes and also schizophrenia. And similar studies were also done in different rodents. They played with the diet and saw that it affects the next generation. However, in most of these studies, people studied only intergenerational effects, not transgenerational effects. The difference between an intergenerational effect and a transgenerational effect, that an intergenerational effect can result from direct exposure of the progeny to the trigger. So, for example, in the case of the Dutch winter, the kids that were examined were in utero, inside their mother, while the mother was starved. So you can say that they were directly affected. Not only that, since the babies in utero already had their germ cells, you can say that 2 generations down the road were directly affected by the starvation. So it's not really inheritance. Only if you continue to the F3 for an additional generation, you can say that you're actually examining a generation that wasn't exposed to the starvation itself. And this is what we did with C. elegans. Moreover, in these type of studies in higher organisms, in mammals, it's hard to separate nature from nurture because these mammals are genetically different, even if it's inbred lines. And also because it's hard to isolate environmental effects or effects of education. In worms, it's very easy because you grow them on food or without food, and you can just bleach them and examine them, and there is no education. Moreover, the worms have a very dedicated and important program to deal with starvation. So if worms hatch from the eggs on a plate without food, they immediately arrest their development at the L1 stage. And when they do this, even if they arrest for 15 minutes, they have gene expression of changes of more than 27% of their genes. So it's a massive change in gene expression, which is bigger than the changes that occur in gene expression throughout the entire course of the development of the other stages. So it's a massive program shift that they do when they are starved. And what we did is we took worms that were well fed, and then we let them hatch on plates with food or without food. Food for these worms is E. coli bacteria. And then we sequenced small RNAs and mRNAs from the parents. And then we moved the progeny to different plates that have food on them. So all the progeny in the 2 groups are well fed. And we sequenced mRNAs and small RNAs from them as well. We followed them for 3 generations, so a true transgenerational effect. And we examined the gene expression changes and the small RNA changes. And what we saw is that the heritable small RNA pools of the descendants reflect their grandparents. So there's actual changes of small RNAs that are enriched in small RNAs that target nutrition-related genes that persist for at least 3 generations, and that these changes change the life expectancy of the worms. So if worms that descend from starved ancestors live longer. Other people studied this further and discovered additional changes. For example, there are changes in the developmental timing, and there are also changes in the capacity to withstand or survive a more difficult period of starvation. We starved the worms for only 6 days, which is a period where they all survive the starvation. You can starve them for 10 days and they all survive, because we didn't want to select the survivors. We just wanted to starve them and let everyone survive. But what people have shown that if you then examine the progeny and you starve them for longer periods where some of them die and some survive, the descendants of the starved individuals survive better. So this sounds adaptive. Other people studied the viral infections after us and also starvation, different types of starvations, and showed that this leads to transgenerational changes that are mediated by changes in small RNAs. People, including us, showed that also changes in temperature lead to transgenerational changes. And recently, a few labs have shown that exposure to pathogens, to pathogenic bacteria, also lead to transgenerational changes via small RNA inheritance. So it seems that different types of environments lead to transgenerational changes, but it's clear that not every environment does it. Some environments do not lead to transgenerational changes. For example, we studied growing worms in liquid, and we saw that while the immediate descendants are different, are longer, this is only an intergenerational effect, and after 3 generations, it goes back to normal. Okay. Now, I think that this is all very interesting, but normally when you think about memory, you think about memories that are made in the brain. Memories that are made in the brain are different and more sophisticated. And I think it's the most fascinating thing is to ask, can changes that start in the brain transmit transgenerationally? The reason that, if you go a little bit deeper into why this is so interesting to me, is that there are 2 reasons. First of all, the brain is better than any other tissue at coordinating responses, bodily responses across the body, internal changes and also external changes, to make calculations. And the second is that the brain, unlike any other tissue, can plan and think ahead. So it can actually, potentially, if it can transmit information between generations, it can plan the fate of the next generation, which is kind of crazy if you think about the implications, and goes around this teleological trap of epigenetic inheritance or inheritance of acquired traits that is so problematic. So I very recently, just a week ago, published a theoretical paper about the boundaries of epigenetic inheritance or inheritance that start in the brain, and asking whether brain activity can transmit transgenerationally and what type of brain activity can transfer in which organism. So you're welcome to read it. But we also studied it experimentally. And but before I get to our studies, I'll go back to this paper that I started my talk with about imprinting. This was the first paper that suggested it, but as I told you, there was only one figure, and it was a skin, a drawing, not actual data. And also, this is a very difficult assay because imprinting is something complex. We wanted to study something that's a very robust and that you have even fluorescent markers that you can follow, something like this. People studied transfer of memory between generations and also between individuals in the 70s and 80s in very controversial studies that are considered to be wrong. And they really buried this question for many years. I will tell you just a little bit about it. So in the 70s, there was a famous researcher called James McConnell, and he studied inheritance in Planaria. Planaria are these flat worms, very different from round worms, C. elegans, separated by millions of years. They are more complex. They are bigger. Also, they have big brains with lobes and everything. And what McConnell said is that, so before McConnell even, I would just say Planaria are really good at regeneration. So you can cut off their heads and they will grow new heads, new tails. They can regenerate their body in a few days. You can even cut them to hundreds of pieces, and each piece will regenerate to a new worm. And this is they're excellent models for regeneration. What McConnell said is that he can teach these Planaria certain things, like navigate in a maze, all kinds of memories. And then it can chop off the heads of the Planaria and wait until the tails grow a new head. And he said that the new heads, the new brains, will retain the memories that the previous head acquired. Not only that, McConnell said that he can teach a Planaria something, grind it, and feed it to another Planaria because they are cannibalistic, and that these naive Planaria will acquire the memory via feeding. This all sounds totally crazy. Indeed, people didn't believe him, although some people did replicate his work, and many people didn't believe him, but it was the least of his concerns because he was targeted by a terrorist called the Unabomber, which sent letters to all kinds of people, including many scientists, and he was exploded. So his line of research stopped prematurely before we really could figure out what's going on. There were follow-up experiments with people who did these experiments in rats and all kinds of organisms that were later found out to be not true. And not necessarily cheating, but just there were methodological problems. So this was considered to be completely wrong for many, many years. And I talk about it in this new review that I wrote. However, recently, a few years ago, a group in Boston, this was the lab of Mike Levine and the postdoc who did it is Tal Shomrat. They replicated some of McConnell's experiment about the chopping of the heads. And they did it using a very sophisticated system that films the worms as they learn and trains them automatically. So perhaps there's something there indeed. They didn't repeat these experiments where where McConnell fed worms with other worms to transfer the information via food. However, I will add that although this was in the 70s, McConnell suggested that the molecule that transfers the memory in food is RNA. And this was long before regulatory RNAs were known. He separated the food to different fractions, so just the DNA, just the RNA, just the lipids, proteins, and so on. And he said that the fraction that transfers the molecule, the memory, is RNA. He was a little bit of a joker, so he published some of his experiments in a journal that he published with jokes and stuff like that. And this also reached the general public and became very, very popular in the just, non-scientific areas. So, for example, there's a comic books about it. There's even a Star Trek episode about this. You see here, the two characters, and they're talking about the memory of RNA, about mechanisms of memory, and one character explains to the other that that memory is encoded in chains of RNA molecules. This is an episode from 1984. So everyone knew about it, but it was considered to be completely wrong. So not because of that, but just because we studied transgenerational memory and because we studied the mechanisms of shuttling of RNA between different tissues, we wanted to know whether in C. elegans, which is a great model for studying this, neuronal memories can transfer between generations. So what we did is, and this was published not so long ago, we took worms that are defective in small RNA production, in the production of endogenous small RNAs. These mutants are called RD4. And as I mentioned before in the talk, RD4 is a double-strand RNA binding protein, which is crucial for the synthesis of certain endogenous small RNAs. And then we rescued RD4 activity. We expressed RD4 specifically in a single copy only in the nervous system of the worms using a pan-neuronal promoter. And we validated that we express RD4 specifically in neurons using literally 6 different techniques. And then in the next generation, we isolated worms that lost this rescuing constant. So they are again RD4 minus mutants. And we sequenced small RNAs from them. And we identified small RNAs that are made by RD4, which could only have come from the ancestor's brain, because only the ancestor's brain created small RNAs that are dependent on RD4. And we also, in this paper, developed for the first time the capacity to sequence small RNAs from isolated neurons. We sequence small RNAs from isolated neurons also of wild-type worms and also from the germline to track the small RNAs that are changing across the body and across generations when you manipulate the production of small RNAs just in the brain. And we found that there are small RNAs that are transgenerationally inherited in response to changes of small RNAs in the brain, and that their inheritance depends on an argonaut, the protein that physically carries small RNAs, that is called HRD1, heritable RNA deficient 1, which is required only for RNA inheritance, but not for RNA within the same generation. This is one of the many argonauts that C. elegans has. C. elegans encodes for 26 argonauts. The genome encodes for 26 different argonauts, these small RNA binding proteins, 19 of which are real genes and not pseudogenes. This one argonaut is needed for this inheritance. We also studied a little bit the mechanism of how the information moves from the neurons to the germline, but there's still many things there that we don't know, and we are studying this now. Importantly, the transfer of information from the nervous system to the next generation is required for controlling behavior transgenerationally. So we found that RD4 mutants, these RNA mutants, are defective in chemotaxis, in finding food in slightly restrictive temperatures, so 25 degrees and not 20. They can't find food, which is crucial. However, and this is unlike wild-type forms. However, if the RD4 mutants derive from ancestors that had RD4 expressed just in the nervous system, so ancestors that could make small RNAs in their brain, then the capacity of the descendants to do RNA, to do chemotaxis, to find food, is much improved, which means that using small RNAs, the ancestors, the grandparents, can transfer the capacity to look for food to the descendants. Moreover, we found that this is mediated by the transgenerational silencing of one gene, which is called SAGE2, which is regulated by heritable small RNAs. Yeah. Okay, and we are now studying more responses that can regulate behavior transgenerationally, and the mechanisms of how the brain communicates with the next generation. Okay, the last thing that I will show you in the last 5 minutes is that small RNA inheritance, transgenerational epigenetic inheritance, follows specific rules. It's not just passive passing down of information. And that the rules of epigenetic inheritance are different from the rules of genetics that we know since Mendel discovered them more than 150 years ago. And the rules of genetics basically just reflect chromosome segregation, meiosis. Epigenetic inheritance follows a different set of rules. People found, and this is very replicable, that if you start an RNA response just by exposing the worms to double-stranded RNA, then the effect lasts for approximately 3 to 5 generations in most of the worms. Some of the worms silence it even longer. But in the population, most of the worms will stop silencing the gene after 3 to 5 generations. And originally, just like people assumed normal inheritance works through blending before Mendel, people assumed that the process, the heritable silencing is the result of dilution or blending. So the parents make certain small RNAs, and these small RNAs are diluted across generations until the effect stops. However, we knew this can't be true because every worm produces something like 250 babies. So after 4 generations, the dilution factor would be 250 times 250 times 250. That adds up to 4 billion after 4 generations, completely homeopathic and could never work. And plus, we knew that there are mechanisms for amplification of small RNAs. I told you that we found that this gene RF1, this RNA-dependent RNA polymerase, is needed for transgenerational silencing. What happens in C. elegans is that there are different types of primary small RNAs, endogenous siRNAs, short interfering RNAs, piRNAs, ribosomally associated small RNAs. All these small RNAs do not lead to silencing. What they do is they recruit to the mRNA templates, the RNA-dependent RNA polymerases, and these lead to the amplification of much more abundant small RNAs, which are 22 nucleotides long and start with the G, which are the small RNAs that actually get inherited. So if the worms can amplify more and create more and more small RNAs in every generation, why does the response ever stop? That's the question we need to ask. And what we thought is that it stops because it's just not beneficial to remember everything that your parents did. You want to retain only the helpful and relevant information. And indeed, we found that it's not passive dilution. What happens is that there's a negative feedback loop that closes down, or shuts down the inheritance after 4, 5 generations. And this is, we call this the transgenerational timer. And we identify genes that we call MOTEC genes. MOTEC in Hebrew, it means sweetheart, but the acronym is Modified Transgenerational Epigenetics. Genes that make up this clock that times the duration of the heritable response. And if you mutate different MOTEC genes, you get worms that inherit RNA for longer or shorter duration. There are some MOTEC genes that never forget. They inherit the RNA responses for hundreds of generations. So it's totally stable. Very recently, just a month ago, we published a paper where we showed that the response is also plastic and responds to stress. So if you start an RNA response, typically it will last for 4 generations approximately. But if you stress the progeny, the F1s, in a number of different ways, by starving them or by putting them in high temperatures or by putting them in high concentration of salt, the response will stop immediately, because the worms will say, this is what we imagine, everything changed, I need to readjust my epigenetic inheritance. And this depends on stress signaling pathways. The worm need to know that it's stressed. Otherwise, stress won't reset. Just changing of the environment won't change the inheritance. I will not get into the details of that, but we can talk about it if you want. But we showed that small RNA inheritance is regulated by changes in the chromatin. So the small RNAs move into the nucleus, they guide chromatin modifiers to the DNA, and the small RNA and the chromatin modifications feed back on the creation of additional small RNAs. And in certain mutants that are defective in chromatin modification, for example, MET2, which is an H3K9 mono and dimethyltransferase, RNA inheritance is stable. It never stops because you need these modifications to stop inheritance. In other mutants, so in a triple mutant that doesn't have K9 trimethylation at all, MET2, SET25, SEN32, the inheritance of some genes doesn't happen. Many genes can be inherited, can be silenced transgenerationally just fine, but newly evolved genes will not be transgenerationally silenced. And this is probably an anti-transposon or an anti-foreign DNA type of mechanism. We also showed that small RNAs get sorted and that small RNA is regulated in these liquid-like germ granules that reside outside of the nucleus, which process the mRNA and choose mRNA that will be subjected to transgenerational silencing. And disruption of the morphology of these germ granules leads to transgenerational epigenetic changes that last for many, many different generations and follow a parent of origin type of thing, where it's different whether you manipulate them in the mother or the father. And we noticed a while ago that isogenic worms, so worms that have the same genetic information, show a different type of response. Some of them will silence the target very strongly. This is true when it comes to viral silencing, but also of other genes, while some will silence the virus very weakly. So there's a great variation, although the worms are genetically identical. And we didn't know why this is the case. So recently we studied this variability by following lineages that are composed by tens of thousands of worms and looking for patterns. We search for these ratios in inheritance, just like the Mendelian ratio, 3:1 or 9:3:1 or something like this. And we looked at patterns in the inheritance in populations, and we identified 3 different rules that are unrelated to Mendelian rules that describe how transgenerational epigenetic responses would propagate in the population across generations. So now, using these 3 simple principles, you can understand, you can predict which worms will inherit the silencing and which won't, and for how long. And I will not get into this because it's slightly technical, but and you can read about it because it's published, but we found that there's an active mechanism that creates on purpose variability between different isogenic worms, which is mediated, controlled by this a master regulator of stress, HSF1, transcription power, very famous transcription factor. And the last thing that I will show, this is just 2 slides, is that we're now asking, can an epigenetic effect eventually lead to a change in the DNA sequence? Because imagine you take 2 populations of worms that are genetically identical. One of the populations is subjected to stress, the other one is not. Let's say that the response would transfer transgenerationally and will be adaptive. Okay, so the population of these descendants, the descendants of the worms that experience the stress would propagate more, it's adaptive, but in the end, they will be genetically identical. So can evolution actually work like that? Or alternatively, perhaps the epigenetic change can also change the DNA. And this would be giving evolution something to work with, because now there will be also genetic diversity. This is one of the most important things to study. We study it now. And what we showed there recently is this, we pre-printed it, it's under review, this paper, is that when you put the worms in high temperature, the small RNAs in the sperm change. And this compromises the functionality of the sperm for multiple generations, even in descendants that are grown in normal temperature. And when the sperm doesn't fuse with the oocyte, the worm, the hermaphrodites, it makes sperm and egg, since the oocyte is not fertilized, the hermaphrodites start secreting pheromones which attract males. And then the hermaphrodites mate, they outcross more, which leads to the diversification of their genome. Because when you mate, you dilute your genome in half, which means that when you subject the worms to stress, you also affect the genetic materials down the road, increasing genetic variability and giving evolution more raw material to choose from. So this could accelerate perhaps the process of evolution. You can read about it in this new preprint. So this is everything I wanted to tell you, and I'll be, of course, very, very happy to answer any questions. So thank you very much. Oh, I forgot this last slide. Normally, people make the distinction between Lamarckian evolution and Darwinian evolution. But in fact, Darwin believed in the inheritance of acquired traits. Darwin and Lamarck they both didn't know anything about genetics. Okay, what Darwin suggested, and this is a mechanism of a Lamarckian mechanism, a mechanism of inheritance of acquired traits. He suggested that every cell in the body secretes gammules, these vesicles that collect in the germline and get to vote on the constitution of the offspring. And people recently suggested that these heritable small RNAs that we describe are in fact something that is related to Darwin's gammules, to the genetic mechanism that Darwin suggested, which was again considered to be completely wrong for many, many years. And what I think is that perhaps in addition to changes in DNA, which are of course the most important things, there are heritable small RNAs that were always in the background, we just missed them, and they affect the inheritance of many traits. And as you know, when the human genome was published and was finished in 2001, people were surprised to find that humans and worms have approximately the same number of genes, and that corn has many more genes. We know that it's not how many genes you have, but how you use them. And inheritance of small RNAs, transgeneration inheritance, could be one way to achieve sophistication. Okay, so that's everything I wanted to tell you. Please ask any question. And thank you very much. And thanks, of course, to the people who actually did the work in the lab and do the funding sources. Thank you very much.
Thanks a lot, Professor Oded, for this amazing talk. So if anyone has any questions, they can unmute themselves and ask it.
Professor Oded, I have a question. So you talked about like early evolved genes and late genes that were present from some time in the worms. So how does this happen that those genes were not transgenerationally inherited? Is there any kind of chromatin remodeling or something like that? Is there a hotspot where these genes exist? Or
yeah, so that's a great question. What we found, and we were very surprised. So people thought the transgenerational inheritance of RNA requires H3K9 trimethylation, this chromatin, suppressive chromatin marks. And this was assumed based on experiments where a GFP transgene was silenced transgenerationally following exposure to double-stranded RNA. What we found is that if you target a gene, and it's true that if you target any germline gene, in the next generation, in addition to finding heritable small RNAs, you also find these repressive chromatin marks, K9 trimethylation. But what we did is we studied worms that don't have K9 trimethylation at all, triple mutants, where it doesn't happen. And we found that while GFP stops being silenced transgenerationally, the endogenous gene regain expression, even though there aren't any K9 trimethylation modifications. So we said, what's the difference? Then we analyzed it globally, and we saw that the only genes that need to be modified by K9 trimethylation to get transgenerationally silenced are newly evolved genes. And the reason is, so we think of K9 trimethylation as something that marks newly evolved genes for silencing, and the small RNAs can probably propagate because they can self-amplify. Small RNAs are made against these genes when the chromatin mark is there, when it comes to the newly evolved genes. How are the newly evolved genes recognized? That's a big question. We don't know the answer. It could be perhaps because they have a suboptimal sequences for splicing, because they're weird, or because they have other weird things about them. For example, perhaps unoptimal codon usage or other stuff that distinguishes them from endogenous genes. People have seen that genes that get transgenerationally silenced, for example, in their introns, they have less AT-rich sequences. So because they're not there long enough, they look suspicious and they get subjected to transgenerational silencing.
Thank you.
Welcome.
Are there any more questions?
Don't be shy.
Abhinav, there is a question in the chat.
Yeah, I don't see. Oh, now I see it. Is the inheritance only limited to order recognition? No. So the inheritance that we found inheritance to occur in response to viral infections, starvation, temperature, and other people have also shown pathogen avoidance. So there are different types of things that gets inherited transgenerationally in worms.
So there is another question. Are the cannibalism animals more intelligent compared to others? Can they form memory, long-term memory easily than normal individuals?
Yeah, so first of all, the experiments about the cannibalism are extremely controversial, okay? We don't know if that's true or not. And I wouldn't say that they're more intelligent, and we don't know. We don't know. What's the next big question someone asked? There are many big questions. One of them is how does the brain communicate with the germline? Can transgenerational inheritance affect the DNA sequence and evolution? Is it conserved in other organisms? That's probably a very big one. Is it conserved in mammals? So there are many, many questions, mechanistic questions as well. This is just the beginning. I see that there's another question. Is there a threshold up to which I think someone is asking a question, but I can't hear very well.
Oded, one of my classmates is asking, is there a threshold up to which differential memory across generation can accumulate?
We don't know. I don't know. What I can say is that I think there's a limited bandwidth to inheritance. So for example, if you start an RNA response and then you stress the worms, it stops the inheritance of the RNA response. And I think this is because different small RNAs compete over the same mechanism. For example, enzymes that process small RNAs. So one memory, if you wish, if you will, come at the expense of another. So there's a limited bandwidth to inherit, which I think perhaps relates to this question.
Are there any more questions?
I have a question as well, one more question. So besides RNA, do you think proteins are also involved in some kind of memory transfer?
Yeah, it's a great question. In yeast, in different fungi, people have shown, and this was shown by primarily by this started with Susan Lindquist, that prions can also mediate all kinds of mechanisms of inheritance, of resistance. This was shown in yeast. So it's possible that also proteins that acquire a prionic state can transmit memories. And also, it's very possible that different types of chromatin modifications can self-template to transmit information across generation. This was very convincingly shown in yeast and in plants.
There's another question in the chat box.
Is the inheritance only seen from grandparents to grandchildren? You mean up to the grandchildren? No. So some responses can even last very long, from multiple generations, after the grandchildren. So long after, and the response can, of course, can be triggered by the parents, not only the grandparents.
Yeah, so if there are no more questions, we'll end our session for today.
There is another question. So Saptarshi is asking, if we pet wild animal, would we expect that the progeny will behave in the same way or as a wild animal?
Can you please repeat this? I didn't hear well.
Saptarshi is asking, if we pet a wild animal, would we expect that the progeny will behave in the same way or as a wild animal?
Yeah, perhaps if you could write it down quickly.
So the question is, if we make a wild animal a pet, would the progeny of a pet, like if we domesticate it,
if we do, yeah.
So would the progeny would also behave like a pet, or would it behave like the wild animal?
So the thing is that if we tame it by changing its genome, then it could be just regularly inherited. If you tame it by via an epigenetic mechanism, then that depends on whether this epigenetic information can transfer transgenerationally or not. So taming is something very complex. I can tell you about one study of taming in C. elegans. It is very nice. It's a paper from Corey Bagman in Nature from a few years, where when people started working in with C. elegans, they kept two different cultures. One culture of worms were grown on agar plates for 50 years, and the other one was grown on in liquid for 50 years. She found that the ones that are growing in liquid to fit liquid handling stop going into downward crowding and going into this alternative developmental mode, because they are used to the proximity and to being grown in a very crowded environment. But this was a simple genetic change that they mapped precisely to two genes. So it really depends on the mechanism. I see there's another question. Is it possible to implant memories using synthetic RNAs? Yeah, I don't know. There's one example of this, which is very recent from Colin Murphy's lab, where she said that a bacterial RNA, particular bacterial RNA, leads to fear of the bacteria, and that this can be transferred from worm to worm. This is a very new study which showed something like this, and it's only a preprint, but that's what they've shown there.
So if there is no more question, thank you, Professor Oded, for joining us today. And thank you for replying. I was not expecting for this to happen, but it just happened. And thank you for coming. We are very happy that you joined us today, and we'll continue to follow you and your hilarious memes on Twitter. Thank you again.
Thank you. My pleasure. Bye-bye.